Ever wondered how aspirin works? To understand aspirin, which is an enzyme inhibitor that enables cell regulation, we first need to understand how enzymes work. Enzymes contain an active site where a specific substrate, or reactant, can bind to the enzyme and undergo a reaction to produce products. Using an analogy, let's say that you're baking chocolate muffins. The muffin batter, which is made of different ingredients, would represent the substrates. The oven would represent the active site of the enzyme, and the baked muffins would represent the products. So what are enzyme inhibitors? I mean, it does sound like a complicated term, right? And how are they connected to cell regulation? Cell regulation involves controlling how much of certain substances is produced in order to maintain balance in the body, which is called homeostasis. Cells produce three types of inhibitors to limit how much product is formed by enzymes. Inhibitors can bind to the active site instead of a substrate, which is competitive inhibition. Some inhibitors can bind to an allosteric site, which is another site on the enzyme. This changes the shape of the active site, which prevents the substrate from binding to the active site. This type is called allosteric inhibition. Both of these types of inhibition are reversible because only weak bonds are formed between the enzymes and the inhibitor. However, other inhibitors form covalent bonds with the enzyme, which permanently alters the shape of the active site. One example of this form of inhibition is aspirin, which is an irreversible inhibitor that stops pain. Wondering why aspirin is needed in the first place? Well, when there is tissue damage, fatty acids that contain arachidonic acid are cut off from the plasma membrane by an enzyme called phospholipase A2. Then, an enzyme called cyclooxygenase, or COX for short, creates a ring in arachidonic acid, the substrate, and adds oxygens to form prostaglandins, the product. Prostaglandins send signals to the hypothalamus in the brain to increase the body temperature in order to prevent pathogens from surviving in the body. The higher temperature also damages the body's own cells, which is what causes pain. Prostaglandins also stimulate immune cells to cause inflammation. So now we are finally back to our original question. How does aspirin work? Well, aspirin prevents the formation of prostaglandins by inhibiting COX enzymes irreversibly by forming a covalent bond with the COX enzyme. This changes the active site's shape and prevents arachidonic acid from entering the active site, meaning that prostaglandins can no longer be produced. Because prostaglandin synthesis is stopped, aspirin relieves pain, fever, and inflammation that you may be experiencing.